Hi everyone, my name is Veselina and I'm a postdoctoral researcher in the Fosin Lactose Labs, and today I'll talk to you about nerve cell repair. First, I'd like to talk to you about two conditions. One is spinal cord injury. As you know, the spinal cord connects the brain to the rest of the body and the organs, and when damaged can have permanent consequences. The other condition is called glaucoma and affects the nerve that connects the eye to the brain and that processes visual information. What's common between these conditions is that no treatment exists at the moment, and they can affect anyone at any time. What's also common is that the central nervous system cells in the spinal cord and in the optic nerve do not self-repair after injury. So if you look at this graph, you can see that early on in development, all mammals and the humans are very regenerative. This is the embryonic to fetal transition, and afterwards they become non-regenerative. This is not true of lower organisms such as nematodes, worms, and zebrafish. Why does this happen? So we think one reason why in humans and mammals there's a decline in the regenerative ability is because there is an evolutionary decline in the transport of growth molecules. So if you imagine this is a nerve cell that's growing from early on in development, it starts growing these long processes that connect it to other nerve cells. When the cell is young, early on in development, it has all these yellow growth molecules all over the cell. But when it grows, these molecules become confined to this part called the cell body, which is kind of like the powerhouse of the cell. And there is no growth molecules in this long process which connects it to other cells. And this is because this process has grown, reached the target cells, and it doesn't need these growth molecules there anymore. We believe this is one reason why these processes cannot regenerate after injury. There is tons of research in the field that targets regenerative processes, and it's actually very successful. However, a lot of these papers are published in animals, and they target processes that are really essential for proper function of the cells. So we can't really target those in humans because they lead to side effects such as tumor formation and so on. Furthermore, a lot of these papers, although they show repair and regrowth, they don't really have any functional consequences. So basically, these cells do not actually form their initial connections. They don't actually function properly like they should. During my PhD, I focused on this fancy protein called protrudin. Why? Well, protrudin, when you add it to cancer cells, such as these ones, make them grow these processes very much like those long protrusions from the nerve cells. And the other reason is that protrudin is actually part of this machinery that carries these growth molecules that I told you are absent from the long processes in adult nerve cells. Well, I told you this process goes wrong in adulthood. So what we try to do is genetically engineer the protrudin molecule to make it more active and therefore move more of these growth molecules into the nerve processes, and hopefully that will help them regenerate better. So what we did first is we added the molecule to cells in a dish from animals, and here you can see they grow these long processes. And then we used the laser to cut a tiny bit of this process and watch it overnight to see whether this repairs itself or not. So you can see when there is no intervention, this process just stays cut. It doesn't really grow, and this part dies off. However, when we add the active version of our protein, these processes start regrowing. And this is one of the best effects we've seen in a dish in our lab. So we decided to see whether this is actually true in an intact organism, and we used the mouse as a model. And together with Dr. Craig Pearson, we injected the protein. This is an eye, and these are the nerve cells in the eye, which project these long processes towards the brain. And then we injected the protein, damaged the nerves that connected to the brain, and then watched what happened. Do they repair themselves or not? So normally when there's no intervention, these white dots are basically the nerve processes. You can see they've died off, they don't really regrow. However, when we add our molecule, we see huge regeneration in the optic nerve after injury. And what's more interesting, we tested our molecule in a disease-relevant model, which is the glaucoma model in rats, where we injected the molecule, then injured the eye with a laser, which causes damage to the nerve and also causes cell death in the retina. This is the retina that we looked at, and the little green dots are the nerve cells in the retina. So when you injure the nerve, these cells start dying off. And when there is no intervention, you can see here there's a lot less nerve cells than when we add our active protrudin molecule. This means that our molecule is not only regenerative, but also neuroprotective, so it protects the nerve cells in the brain. And of course, the question is this because there's more growth molecules in these processes? And I wouldn't be telling you this if the answer was no. So this is when we have no intervention, there is almost no growth molecules, and if there is any, they're always moving backwards, and there's like one or two. But if we add our active protrudin, we can see loads of molecules shooting up and down these cell processes. And, sorry. So I wanted to tell you also how I use my CSAR Award. First, I performed a research exchange with McGill University in Canada, where I learned some techniques that we did 3D optic nerve imaging, and I'm trying to bring this back to Cambridge at the moment. And I also went to a conference in Austria on optic nerve, and there I formed some collaborations from which really exciting results also emerged. So I'm just going to quickly show you these before I finish my talk. This is people that I met in Austria, and what we did with them is, again, injected our protein into the eye. We actually took out the retina, which on itself, it's an injury. And normally what you see is, this is before the injury and this is after. You see a lot of cells start dying off. These are the nerve cells of the retina. And you can see there's a massive drop in the amount of cells in the eye. However, when we add our molecule, either in its native form or in its active form, we see complete neuroprotection of the retina. This is very exciting for 3 reasons. One is that recently gene therapy has been shown to be successful in the eye because it's a very accessible organ. You can target genes in the eye and you can actually reverse blindness. And we're really hoping to move further into testing our molecule into a human system. Second of all, we discovered, I didn't talk about this, but if anyone is interested after the talk, we discovered numerous pathways that are novel and involved in this regenerative process, and we can actually look for new drug targets to treat axon injury. And finally, I'm currently testing the molecule's efficacy in a mini human brain system, which is made of embryonic human stem cells, and we call these cerebral organoids, and we can test whether our molecule could be effective in a human system. And with this, I'd like to thank you for your attention, and to thank everyone in my lab, especially Professor Fawcett and Dr. Richard Eva, and all my funders, and I take any questions.
Well, thank you, Veselina. Absolutely fabulous. Question at the front here. Yep.
Oh, sorry, I put my hand up after somebody said. It's a question in the front, sorry. Protrudin is the protein you're focusing on. Are there other proteins that do the same job and that you may later use a combination of them? So how many similar proteins are there doing the same job?
The answer to this is we don't know, the short answer, but we are looking for this, and we think there might be some similar proteins with similar functions, and it will be really interesting to target them as well in combination. There is similar proteins also acting in the dendrites of the cell. So the axon was the long process that connects the cell body to another cell, but there's these tiny processes on the cell body where other cells connect onto, and there's similar proteins acting there. So maybe a combination of both could be beneficial, but we haven't really tried out these candidates yet.
Is it single digits or multi-digit numbers of proteins that would be doing a similar job?
We think it's a small number. At least the ones we've looked at that have similar structure is a very small number. So, yeah.
I noticed that you're adding the protrudin before the injury. Have you tried doing it afterwards? Because in terms of normal injuries, that's normally the sequence.
Yeah. Yeah, no, we haven't tried that. This is on our list. We're currently also trying this model in a spinal cord injury model, and we're doing a before and after treatment. So this will be able to tell us more whether it's going to be beneficial after the injury.
Thank you very much for the talk. I was just wondering if there are any mutations that are associated in humans for this, and if so, do they express a phenotype at all?
Yeah, so there's been one family discovered in Germany with a single mutation in the protrudin gene, and that leads to hereditary spastic paraplegia, which is a disease, a motor disease. Basically, the neuronal processes in your limbs die off, and you start getting motor phenotypes, and that's very much connected to the transport of growth molecules inside nerve cells. So. But it's quite a controversial mutation because it hasn't been found in any other family, and the mutated genes didn't really cause any disruption in cells or in animals, so we're not really sure. It hasn't been confirmed, basically, but it's the only one that's been found.
Last one.
Thank you very much. When you give this treatment, you get a repair to a damaged nerve connection. Does it sometimes make new wrong connections? That is, if you want to repair an optic nerve, you want it to make the right connections and not start connecting everything to everything else?
Yeah, so I briefly mentioned this, but our biggest hope with this particular molecule is because it brings these growth molecules towards the end of processes. We hope that these growth molecules can kind of signal from the environment to basically guide their growth towards the right connections. Because these preliminary experiments were done with really short time points, they didn't really manage to quite reach the brain regions. So now we're repeating the experiments with a much longer time point and then looking for exactly that. Is it forming the right connection? Is it improving function, which is what we're really hoping to see. And we believe that this might be the case from our preliminary experiments, but it's still quite early on in the process.
Veselina, thank you. That was absolutely fabulous.
Thank you.